Thank you. Hi, I'm Grant. I work with Professor Alonso Moroni at CSL. And today I'm going to be presenting on the metagenomic binning problem. So what is metagenomics? Metagenomics is the study of genetic content of communities of microbes in the environment. So that environment could be the human microbiome or soil or the surface of the ocean, etc. And these samples contain hundreds of species of bacteria, viruses, etc. And what researchers do is take all of the DNA in a sample and sequence all of the species' DNA. So each species has a genome, and you kind of concatenate all of that and sequence all of it. And you get millions of short DNA reads. From there, they're assembled into much longer sequences. So reads are roughly 100 base pairs long, and these contiguous sequences called contigs are around 10,000, somewhere in that range. And the way that you assemble that is just by overlapping reads and merging them to get these much longer sequences. So I'm going to be using this term contig a lot throughout the presentation. So all it means is a long string of DNA. It's not a genome, but so any given contig comes from a single species genome, basically. And metagenomics, the job is to find what this species profile is. Not sure why the laser's not working. So yeah, okay, anyway, the job of metagenomics is to build this species profile. And this profile provides insight into the diversity and health of a sample. So if your sample's from the human microbiome, you expect a healthy sample to have a diverse set of species. Specifically, the metagenomic binning problem is the problem of taking these contigs and clustering them into bins, where each bin corresponds to a species. And using some database of known species, you can assign the species to each bin. Okay? And then from there, it's easy to build this species profile. Okay, the harder part is actually clustering in the first place. So how do we cluster? Well, the state-of-the-art methods use something called the Tetranudeotide Frequency. It sounds fancy, but all it is the frequency of four-letter strings across a sequence. So how many times you see AAA and how many times you see AAT, etc. And it's been shown empirically that this is remarkably consistent across a given species genome. So intuitively, two contigs, which come from the same genome, likely have very similar TNF distributions. So you can compare them in this way, where, for example, AAA, you roughly have similar frequency in both contigs. So this contig, or these two contigs, likely come from the same species. So how do we actually do this a little more formally? You can't simply compare these distributions directly because, okay, let me backtrack a sec. So we model these contigs as being generated by a species TNF. So a given species has a TNF, and it basically generates this contig. But you can't generate these contigs IID on this distribution because consecutive four-letter strings overlap in a sequence. So what's the natural model from here? Well, we model it as a Markov process. So for example, in TACG, the G depended on TAC, the previous three. So each is a third-order Markov process, where the TNF is the stationary distribution of the process. Okay, let's define our problem a little more formally. We assume we have M species in our sample, and each species corresponds to a third order Markov process, pk. And these species are unknown. So if we knew them, then the problem would be a lot easier. The harder part is finding out what these distributions are. So the M species generate n contigs, okay? So where each contig has an equal likelihood of having been generated by any given species. We want to know the following question. When can we perfectly bin all contigs as the number of contigs n goes to infinity? And right, so what do we mean by perfectly bin? Well, we mean so what do we mean? We mean that we want to generate these bins such that each bin perfectly corresponds to which species generated which contigs, okay? And you might think at first, well, if we have an infinite number of contigs, you're likely to make an error on at least one of them. But if we have the length of each contig growing with n, then each contig gives more information about which species it came from, okay? And specifically, we show that this regime where L is proportional to the log of n is an interesting one in that this constant L bar controls when and when you cannot perfectly bin all contigs as the number of contigs goes to infinity. Okay. Our main result is that as n goes to infinity, perfect binning is achievable if and only if this constant L bar is greater than 1 over the minimum Chernoff information between two species. This is a lot to process, but all this is saying is that this constant L bar, this threshold for when you can and can't perfectly bin, depends on the two closest species in TNF, in the 4-letter distribution. Okay. So I'm going to define what the Chernoff information is later, but for now, just think of it as a measure between how close species are. So how do we actually perfectly bin all contigs? What's our achievability scheme? Well, first we need to measure the underlying species distributions. We need to get a good estimate in order to have some ML rule, which assigns contigs to species. So for each contig, we calculate its empirical 4-letter distribution, its TNF. And we build a graph of the contigs, where an edge is formed if the L1 distance between two contigs in TNF is less than some value epsilon. And we're going to start epsilon off at the minimum possible distance. So there's a tiny little edge right there. And then we're going to increase epsilon until we see M large clicks form. So you increase it, and M because we want each click to correspond to a species. And okay, why do we want the size of the click to be greater than this value? Well, n over m is the expected number of contigs per species. There's n contigs and m species. And alpha is simply a slack variable. So you can't expect each species to perfectly produce n over m. So the goal here is to estimate the underlying species TNFs. So what we do is average the TNFs of each contig in each click. And this gives us an estimate for the distribution. And it can be shown that in the limit as the number of contigs goes to infinity, that these actually converge to the true distributions. Okay, so from here out, we're going to basically assume that we have the underlying distributions. From here, how you assign a contig to a bin is simply by the ML rule. We have the distributions that generated the contigs, and we simply assign it to the maximum likelihood probability distribution. And this is a classic information theory result where the maximum likelihood corresponds to the minimum KL divergence between two distributions. But the interesting thing here is that these aren't IID, so this is actually, since it's a Markov process, this is actually a conditional KL divergence. Okay, so it's a little trickier because here you need to take into account initial conditions when you don't need to do that for an IID case. And with a little algebra, you can show that this conditional KL divergence is equal to the normal KL divergence between the TNFs minus the KL divergence between the three-letter distributions. Okay, so now that we have this ML rule, let's find what our probability of error is in this and show that it goes to 0. So let's look at the binary hypothesis test between two species. Okay, so we just showed that this dividing line where a contig on this side is assigned to P2 and a contig on this side is assigned to P1 is where their conditional KL divergences are equal. Okay, so let's assume we have a contig generated by P1. Intuitively, the farther away it is from P1 in KL divergence, the less likely it was generated by P1. So here, it's still assigned correctly, but eventually you're going to get to a point where you're incorrectly assigning the contig to P2. Okay, so this point where it's still likely, somewhat likely to be generated by P1, but it's incorrectly assigned is P star. We call it P star. And this distribution is essentially the bane of the test because most of your errors are going to come from where P1 is generating a somewhat likely contig, but it's incorrectly assigned to P2. Okay, so this error overall is P1 of this region script P2. Okay, so another classic information theory result is that Sanov's theorem tells us that the error exponent in this hypothesis test is the KL divergence between P star and P1. But here, again, it's interesting that this is actually a conditional KL divergence. It's not nearly as straightforward for this case. And this is what we define as the turnoff information between P1 and P2. So it's symmetric. If we were to do the same thing with contig generated by P2, then P star would be in the same place and the turnoff information would be the same. Right, so how does this prove that L bar needs to be greater than 1 over the minimum turnoff information? Well, the turnoff information, the minimum turnoff information is essentially the error that's going to go to 0 the quickest, or the slowest, sorry. So it's kind of the limiting error exponent. And intuitively, when L is large, when you have more information for each contig, then it's easier to bin compared to the number of contigs you have. So if L bar is really big, then it doesn't matter that you have a growing number of contigs because you'll have more information in each contig. So this is roughly our error. It's n contigs, and it's e to the minus c min because c is the error exponent. So if we put the n up in the exponent, and since log of n is actually equal to L over L bar, we can pull out an L, and we get this 1 over L bar minus C min. And since we need this exponent to be negative, that gives us our result, that L bar must be bigger than 1 over the minimum Chernoff information in order to perfectly bin all contigs. OK, why do we care about all this? Well, in practice, what the state-of-the-art tools use is just a Euclidean distance between TNFs in order to get a similarity between contigs. But we just showed here that the conditional KL divergence is a better metric. It's a more principled approach. And so what I did was take two species with the coolest names I could find and did this binary hypothesis test using contigs from the actual genomes of these species. So I